it's a great pleasure and honor to speak in this seminar series. So today I will talk about properties of memory networks with excitatory-inhibitory assemblies. So since the 1950s, there have been two influential concepts to describe memory formation and retrieval, and these two concepts are neuronal assembly and attractor states. So classical views suggest that when you memorize information, like for example, a particular smell, the connection between coactive excitatory neurons will strengthen. And so this subset of highly connected excitatory neurons will form an excitatory assembly. So these learning-induced changes in connectivity will affect the dynamics and geometry of activity. So we can represent the neural responses evoked by some external stimuli in population state space. And so here in this space, each axis represents the activity of one neuron. So here I depicted one, as an example, the representation of two odors. And so the geometry of activity would correspond to the organization of this odor representation in state space. So before learning, these representations are diffuse, and this reflects the variability of olfactory stimuli or other sensory stimuli. And so we know from previous theoretical work that the formation of an excitatory assembly will lead to the formation of an attractor state. So this is what you see here on this little toy cartoon. So let's say in this example, the mouse memorizes the smell of lavender, which is here the blue point. And so the activity in response to lavender, as well as to olfactory stimuli similar to the smell of lavender, will converge to one point in state space. So looking at the geometry is interesting because it informs us about the computational properties of the network. For example, attractor states favor robustness to noise and generalization. So even if the sensory input is only partial, the memory can still be fully retrieved. However, this comes at the cost of more fine discrimination and flexibility, which means that if the mouse, for example, encounters a different variety of lavender, the brain activity will converge to the stored pattern of the lavender the mouse encountered first. So this is the classical view. However, there are experimental and theoretical findings that suggests that these two concepts of assembly and attractor states need to be in part revisited in the context of long-term memories. So first, the experimental evidence for attractors during memory retrieval remains weak. And also, if you think about it, the increased recurrent excitation within these excitatory assemblies will lead to activity amplification and eventually to network instability. So there is a well-suited mechanism to counterbalance this strong excitation within assemblies. And so, theoretical work has shown that this mechanism is specific inhibition. So specific inhibition can stabilize activity when learned inputs are presented without affecting the response of novel inputs. And so I will illustrate this a bit more, so this statement a bit later in the talk. So earlier work also has focused on the structure of excitatory connection and plasticity of this excitatory connection. However, we have more and more increasing experimental evidence for plasticity also at the input and output synapses of inhibitory neurons, and also evidence for specific excitatory-inhibitory circuit motifs. So this is why we make the hypothesis that memories are encoded not in excitatory assemblies, but in excitatory-inhibitory assemblies, which we will define here as groups of both excitatory and inhibitory neurons, which become highly connected after learning. And so the broad question we are interested in is, how does this structured connectivity affect memory function? And to address this question, we will build a data-constrained model of a memory network. And so the data we will use is from DP. which is the zebrafish homologue of olfactory cortex in mammals. And so DP receives input from the olfactory bulb, which itself receives input from the olfactory epithelium, where odors are detected. So why do we use DP to search for EI assemblies? Well, first, DP is thought to play a key role in the storage and retrieval of memories, and more specifically olfactory memories. So it is a highly recurrent and plastic network. Furthermore, it exhibits excitatory-inhibitory co-tuning. So this means that if you look at this little scheme here, if you record in one neuron the inhibitory currents and the excitatory current in response to different odors, you will see this nice co-variation. So this suggests that there is some specificity in the connectivity of inhibitory neurons. So I want just to also briefly mention that EI co-tuning has also been observed experimentally in mammalian cortical brain areas like auditory cortex or visual cortex. So now we built a spiking neural network model of DP, which consists of 4,000 excitatory neurons and 1,000 inhibitory neurons that are sparsely connected. So the parameters of the model are taken from the literature when available. For example, we know that the resting conductance of the excitatory neurons is around 1 nanosiemens. And the remaining parameters are determined to reproduce the following measured observables, which I put here in this little box. So here we know that upon odor presentation, so each odor will activate a different pattern of activity, and the mean rate is of the order of 1 Hertz. Also, upon odor presentation, DP enters a balanced EI state, which means that excitation and inhibition are both strong and establish an average membrane potential near spike threshold. And lastly, a large fraction of the excitatory inputs consist of recurrent inputs, so from inputs from other DP excitatory neurons. So now, how do we simulate the odors? So we shortcut the olfactory epithelium pathway, and we define our odors as activity patterns in the olfactory bulb. And as you can see here in the plot on the right, each odor is defined by a specific subset of cells whose firing rate will increase during odor presentation. And now if we feed this input into our DP model, we get this representative response of excitatory neurons here on the left, and as well as these representative traces of membrane potential and synaptic currents. So now, as said, we are interested in memories, so we need to store these memories in our network. So we start with a random network, and then we create assemblies. So to do this, we rewire the connections following a predefined set of rules. So here, let's say, for example, that we want to learn this magenta odor, which is represented by a subset of activated olfactory bulb cells. And so we identify the subset of excitatory neurons in DP that are highly connected to these activated olfactory bulb cells. And then we increase their connectivity. And so on average, this subset of cell contains the most active neurons. And if you think in the context of synaptic plasticity, it would be the most likely neurons to increase their connections. So now to avoid instability, we need to increase inhibition. So to create excitatory and inhibitory assemblies, we specifically increase the connections between a subset of inhibitory neurons and these excitatory assembly neurons. And so these inhibitory neurons are chosen such that they are the most highly connected to these excitatory assembly neurons. So I want to mention here that each assembly is relatively small relative to the total size of the network. So it comprises around 2 to 3% of the total neuronal population. Also, we increase connectivity in such a way that the mean firing rate in response to the learned odors is of the order of 1 Hertz and similar to the mean population rate we see in random networks. So here on the bottom, you see an example raster plot. And so odor 1 would be a novel odor and odor 2 is the learned odor. And you can see here, for example, this, if you see my mouse, this would be an assembly neuron, and you see a modest amplification of activity in this neuron. So now we want to compare these networks with EI assemblies to our classical excitatory assembly networks. And so here we just globally increase the weights from the inhibitory to excitatory neurons. So we have this, again, these excitatory assemblies, but now inhibition remains global, let's say. There is no structure in the inhibitory connection. And so when we increase this inhibitory to excitatory weights, this stabilizes activity. But I mentioned it briefly in the introduction, this unspecific increase in inhibition will affect the activity in response to novel odors. And this what you hopefully can appreciate here in the spike raster plot. So the mean firing rate in response to odor 1 will be divided by 2. So instead of being of 1 Hertz, it will be 0.5. And this affects considerably the detection of novel odors. So now we have a memory network. So we check first that introducing these excitatory inhibitory assemblies will establish EI co-tuning as observed experimentally. So this is what you can see here on the plot on the right. So we measure the correlation in each neurons between the excitatory and inhibitory currents. And you see that it's much higher in networks with EI assemblies than in random and networks with excitatory assemblies. So now we have the model all set, and we can dive more into the scientific question. So in this talk, I want to show you first how these EI assemblies shape the geometry of neural activity. And in the second part, I want to briefly show you how we can unmask these assemblies by unspecific perturbation of inhibition, and how manipulation of inhibition can also reveal novel function of specific inhibitory connectivity. So now, how about the geometry of neural activity in networks with EI assemblies? So to investigate this, we generate an odor subspace. So here a virtual square with 121 odors. So each pixel here would be one odor. And you can just imagine that there's one odor in each corner, and the rest of the odors are just mixtures of these corner odors. We present these odors several times, so we have different trials. And so this generates like a subspace with odors with different levels of similarity. So now remember, in our simulation, I said an odor is equivalent to an olfactory bulb activity pattern. So it means that we can represent this odor square in activity state space. And we can apply a dimensionality reduction to have a new state space where the axes are not the activity of the neurons, but where the axes are patterns of neuronal covariations. So here in this case, we will apply a principal component analysis. And you see that in PCA-reduced space, the activity of the olfactory bulb is confined to a subspace with a squared shape. So now we feed these olfactory bulb activity patterns in our DP model with different types of connectivity, and we then apply PCA on the DP activity patterns. So in the random network, which is here on the bottom left, we also have like a square in PCA reduced space. So the geometry is preserved. Now let's say we want to learn the two corner odors here, so the pink and the violet, meaning we create two corresponding assemblies in DP. So in networks with excitatory assemblies, which is here on the bottom right, we see the kind of classical picture that I mentioned in the introduction. So activity is distributed in 3 locations in state space, and the 2 locations highlighted with the arrow correspond to the attractor states where the activity of the learned and related odors will reside. So now, of course, we want to know what the picture is with networks with EI assemblies. And so here the picture is completely different. So we don't see the emergence of discrete states, but the representation of the inputs remains continuous. And you have these local deformations in the vicinity of the learned odors. So plotting activity in 2PC, so here in 2PC space gives us a good intuition of what is happening. But let's now dive into some more quantification. So first, we can look at the dimensionality of activity. And so dimensionality of activity corresponds to the number of components which would be needed to recapitulate the observed neuronal activity. So here we use the participation ratio, which is a measure that depends on the PCA eigenvalues. So you see that here on this plot, the dimensionality of random networks is relatively high, and the dimensionality of networks with excitatory assembly is relatively low and reflects the discrete attractor states. And now the dimensionality of networks with EI assemblies is kind of in between. So it seems that these EI assemblies constrain neural activity. So now I want to show you that this is local and happening in the vicinity of the learned odors. So to do that, let's cut the virtual square in half and consider only the part which does not contain the learned odors. So here, yeah, the 2 corner odors are novel and there is no corresponding EI assemblies or E assembly. So in this case, you see that the dimensionality of the random networks and the networks with EI assemblies is similar. Now, as expected, if we look at the other half, which contains the learned corner odors, you see that we have again the same picture with the dimensionality of networks with EI assemblies being not as high as the networks with the random networks, but being higher than the networks with excitatory assemblies. So EI assemblies locally constrain neural activity onto lower dimensional subspaces. So now we are going to define the subspace in which activity to one odor is constrained as an odor manifold. So here we have this kind of point cloud manifold, and each point corresponds to a trial of a corner odor. And you can see here also how the learned manifolds, which I highlight with my cursor, are better separated from the other odors. So we can further characterize the geometry of odor representations by calculating the distance between these manifolds. So here we use the Mahalanobis distance because it takes into account how activity is distributed within manifolds. So let me explain a bit. So let's say we want to calculate the distance between the orange or novel manifold and the pink or learned manifold. So here for random networks, I put the learned into quotation marks because to indicate that this is the order which will be learned in the networks with EI assemblies. So we can calculate the distance in 2 directions, either from the center of the orange manifold to the center of the pink manifold, by taking into account the covariation of neurons within the pink manifold, or from the center of the pink manifold to the center of the orange manifold, taking into account the covariation of neurons within the orange manifold. And so if the neural covariance of the two manifolds is different, then these two distances will be different. And so, yeah, for example, here in the case of the networks with EI assemblies, the distribution of activity is not isotropic. The distribution is stretched along one dimension. And so this is indeed what we observe. So in the random networks, the direct independent of direction, the Mahalanobis distance will be similar, but in networks with EI assemblies, it will be higher in one direction than the other. And so this is nice because this is something we can test on experimental data. And now I will show you data acquired and analyzed by people in the lab. So Buhu, Nesibe Temis, Peter Ruprecht, and Benjamin Titze. And so they did calcium imaging during presentation of 4 odors in 2 groups of fish. So one group was a naive fish where there was no learning, and one group was trained fish, in which the two odors were associated either with the presence or absence of a food reward. So in our case, this would correspond to our learned odors if we make the assumption that during this learning there was a change in connectivity and formation of EI assemblies. And so here we see the same trends and what the model would predict. In the naive fish, the Mahalanobis distance was the same independent of direction, but in the trained fish, you have this asymmetry depending on if you look at the distance from the novel to the learned odors or the distance from the learned to the novel odors. So now let me briefly discuss the computational properties of these networks. So we have seen that EI assemblies reorganize odor-evoked activity in neural population state space. So if we think in terms of information encoded, we can think of EI assemblies as retaining information about both the experience of an animal and information about sensory variables. So if you think in networks with excitatory assemblies, this information about the external world or the sensory variables is lost, and each memory formed by an animal is stored as a discrete item. And yeah, I have shown you that in terms of dimensionality of activity, this dimensionality is in between the dimensionality of random and networks with excitatory assemblies. And in terms of computational properties, low dimensionality is thought to favor noise robustness and generalization, as is the case of, for example, of our discrete attractors. If you remember my lavender example in the introduction, if you have a flower with a similar odor, the activity would converge to the stored lavender pattern. And so higher dimensionality is thought to be best suited for discriminating different sensory inputs. So because each point in state space can be well separated from the other points in state space. So in some sense, we can think of networks with EI assemblies as providing kind of a trade-off between generalization and discrimination. So if you are interested in our paper, we show a few examples. For example, we show that in an odor classification task, you have enhanced classification of learned odors, and also that these networks can provide a metric to assess the similarity of inputs to learned odors. So I will give you a very, very quick illustration of this. So what we did is to regress the concentration of the odor against the neuronal activity, because as I said, so in this virtual square, we have four corner odors and then mixtures, which means that we have different concentrations of the corner odors within the square. And what we show is that when you have EI assemblies, you have improved assessment of the concentration of learned odors, but the assessment of novel odors are unaffected. If you're interested, you can have a look at the paper. So this was about the geometry of activity in networks with EI assemblies. And now, in the time remaining, I would like to show you now the effect of perturbations on the activity of these networks. So first, we need to refine the model to take into account new findings about the inhibitory populations in DP. Indeed, Thomas Frank from Göttingen, he characterized two types of inhibitory neurons in DP, which you can see here labeled in green and magenta. So these two populations of inhibitory neurons differ by their connectivity. So the DLX positive population is a feedback, which means that it receives only input from DP neurons, and the 1212C positive population is a mostly feed-forward inhibitory population, which means that it receives input from the olfactory bulb. So now we refine the model and we form assemblies of excitatory and feedback inhibitory neurons. So why feedback inhibitory neurons? Because the feed-forward inhibitory neurons, they don't receive excitatory input from DP, and therefore they cannot control the amplification of excitation by recurrent connectivity within assemblies. And just in case you are wondering, refining this model gives similar results in terms of geometry of activity. So now we partially inactivate either the feed-forward or the feedback inhibitory neurons, and I will abbreviate this PIN on the slide. So as expected, if you partially silence either the feed-forward or the feedback inhibitory neurons, this will lead to increased firing, as you can see here on this trace. So black is without PIN, and green and purple would be during PIN. And you can see on the right plot that there's a critical fraction of feedback neurons which are needed to stabilize the network and prevent runaway activity. So here we stay in the orange range, and instead of looking at the mean firing rate, we calculate the correlation between activity patterns evoked by pairs of odors. So let me first explain what we observed using a kind of very simplified schematics. So we have two odors. So here, odor A in blue and B in brown, and they provide differential input to DP. So the correlation between this olfactory bulb activity pattern is very low. Now, you can see here the activity of the excitatory neurons in DP in response to these two different odors. And the correlation between these two activity patterns is also quite low. Now, if we partially inactivate feedback inhibitory neurons, this correlation will strongly increase. And so this strong increase is what we call runaway correlation. And so why do we see this strong increase? So first, because here neurons belonging to the same assembly received input from both odor A and odor B. And when you have not a precise balance of feedback inhibition, these inputs can be amplified a lot within the assembly because of the increased connectivity within the assembly. And so as a consequence, the correlation between the patterns is in response to odor A and B will strongly increase. And so when inhibition is restored, both the activity, so the activity within the assembly will be lowered and the correlations will be lowered. So this means that these runaway correlations are driven by assembly neurons. But even if we wouldn't know which neurons are part of an assembly, we would still observe only a small subset of excitatory neurons that respond strongly to both odors and that contribute most to the high pattern correlation during this partial inactivation of feedback inhibitory neurons. And so this is what you see here on these plots. You have the firing rate in response to one odor, firing rate in response to the other odor. Each dot would be one neuron. And in the color code would be the contribution to the correlation. And you see here a small subset of neurons which contribute a lot to this runaway correlation. So now these runaway correlations do not occur for all odor pairs, but only for some. And this is what you can see here more clearly when I plot the correlation during PIN as a function of the control correlation, so the correlation when inhibition is intact. Right. And so to convince you that these runaway correlations emerge in the presence of excitatory feedback inhibitory assemblies, here you have the same plot, but for networks with random connectivity. So if you think about it, functionally, these strong correlations impair odor discrimination and memory retrieval, because without fine, precise inhibitory connectivity, even two different odors could generate similar activity patterns in the network, and then this would become much harder to decode. Right, so here you can see better the runaway correlation. So these are the correlation which increase strongly upon PIN. So here we set threshold of the difference between the PIN correlation and control correlation should be greater than 0.25. And here on the right, you see the histogram, which indicates the relative frequency of this runaway correlation. So now, these are again predictions of the model, and we wanted to test them, of course, experimentally. And so to do this, Thomas Frank, he optogenetically manipulated either the DLX positive neurons, which are the feedback neurons, or the 212C positive neurons, which would correspond to our feed-forward population. And as expected, he also saw increased firing during partial inactivation of these inhibitory neurons. But more interestingly, he also observed this runaway correlation when he partially inactivated DLX neuron. And this is what you can see here on the right plot. Furthermore, in his case, also these high correlations were driven by a small subset of strongly active neurons. So these results are in agreement with the prediction of the model. So to summarize, I've shown you two signatures of EI assemblies, so two complementary approaches. So first, a geometric approach. So you have a deformation of odor representations in the vicinity of learned patterns. So this has various computational benefits. And there is a recent experimental data which is in agreement with these predictions. And the second approach, the perturbative approach, where we see the emergence of runaway correlations. And so an important function of this specific inhibition would be to suppress this correlation. And these correlations, we found them in ex vivo DP. So if you are interested, you are welcome to have a look at papers and preprints. So this work here was done in collaboration with my my postdoc advisor, Rainer Friedrich, and our collaborators, Thomas Frank and Friedemann Zenke. I also showed you data from Peter Ruprecht, Bohu, Nessie Betemis, and Benjamin Titze. And yeah, I would like to thank you all for your attention.